So András Palionis, our guest, has worked in IT and biology for well over 30 years. He received numerous awards, and he would just like to bring to our attention this quite big wave of genomic-related data coming our way and our very limited ability to make much sense of it at the moment.
Thank you. Normally, I don't use my full qualifications because 1 or 2 PhD is usually enough. And the third one is a bit scary. But since this is so close to Halloween, I thought I might be able to afford that. You can look me up at the junkdna.com domain. Also, you can email me about issues. And I will make several announcements in this talk. And also, I would like to be as provocative as possible. One announcement is that there is an international hologenomic society being developed. And our question, therefore, is, what is hologenomics? Now, hologenomics is genomics plus epigenomics expressed in informatics. And of course, in a Google setting, this is the key word. Announcement 2 is that the society I established as International Post-Genetic Society is morphing into hologenomic society. The next announcement is that we are going to hold our inaugural meeting. And then if you would like to enroll or enroll the society or sponsor the meeting, visit the hologenomics domain, please. Announcement 5, that there is a HologenTech startup, because if there is a gene making business, then there is Genentech. If there is hologenomics meaning business, there should be a HologenTech. Now, hologenomics is the latest addition to a very long story. Most people don't realize that genetics is over 100 years old. Bateson in 1905 used the word first, and that was just very loosely the science of heredity. Then epigenetics was thrown in by Weddington in 1942, but the concept was redefined at least 15 times. So it's very, very hard to tell what it is. It's basically everything about genetics. Genomics was established as a word by Sanger in the UK in 1970, which is the genes and also the chromosomes. So he tried to bring it all together. Now then epigenomics, not epigenetics, again is some extension on this earlier concept, which is everything about genomics. This is an umbrella term, of course. There is no particular person associated with it. Now, in 2005, when people still believed that 98.7% of the DNA was junk, we established the Post-Genetic Society, which is Genomics Beyond Genes. And in 2006, actually, we had a meeting when we branded junk DNA officially as a misnomer. That happened before the government did it 1 year or 8 months later. And now, trying to bring everything together, hologenomics throws in informatics big way, and it's basically a holistic approach to heredity, which with a message that stop slicing and dicing, let's unite in information theory and deliver by information technology. Most people, when we say IT, we mean information technology. As you can see all through my talk, I will talk about IT1 and IT2, and there is also information theory, which is important for us to develop information technology. Now, a little bit of the history, where we came from. Genomics is information, said as recently as January 2008, Richard Dawkins, who is a very neutral person. He is not a mathematician or physicist. And in fact, he retired 3 days ago from his office. But the story really started the big way from Watson and Crick. Watson retired about a year ago, and Crick passed away in 2004. Now, this is a less well-known figure, Ono, who came up with the idea that 98.7% of the DNA is junk, he said. and he passed away in the year of 2000. Now, the old math said that the genome, the human genome, is about 3 billion bases of A, C, and T, and G letters. And only about 1.3% are the genes. And the functional description was for about half a century ruled by the central dogma of molecular biology. I will talk about it in a minute. And also it was ruled up by this other notion that the two axioms. And this axiom is that actually the genes are very few and the junk is a lot. So if you do the math, then the genome information used to be 40 megabytes. And I hate to say this, that in Google, 40 megabytes amounts to nothing. So it's, the genome baby was a very tiny, tiny thing. Now, the genome baby, if you consider it as information, then, of course, the information technology guys, the information technology guys, they should pick it up. Because if it's information, then they know the means, the mathematical, theoretical, and technology means to pick it up. And this is a not very well-known picture of Anne Wojcicki, because she did pick up the DNA baby by establishing 23andMe a little over a year ago. Sergey Brin helped in. And this technology that they use can deal with 1.6 million letters of ACTG. And essentially what they do is they run a spell checking whether the ACTGs are in a correct order. And if something is wrong, they call it technically a SNP. And that a SNP can be indicative, statistically correlating with some genomic glitches, which may or may not be harmful. You can attach a statistical number like 58% or 23% proclivity if you would develop in your lifetime a particular disease. Now, this I should go very fast through this one. Sergey Brin, the same person who is, of course, very well known in Google, he disclosed on his blog, so it's an open domain information, that actually they checked his genome. And in this gene, they found a SNP which elevates his risk of Parkinson's. Now, therefore, the conclusion is that just by picking up 1.6 million points, the IT already proven itself because there is a direct-to-customers business model. 23andMe works from the customer direct to the company. The doctors doesn't even have to be involved. And also, it is proven by several stories that this kind of checks may help those with genomic risks to prevent disease because they can catch this proclivity before the disease develops. And sometimes there is a lot of time to do something, preventive measures. There are several cases in the literature, not just with this company, but also with others. Now, this tech talk will redefine the risk, actually, by a new information theory IT1 axiom, the principle of recursive genome function. Please look this up in the web. This is a peer-reviewed science paper that was published. I published it this summer. I could not have possibly come up with it until all the kings, the emperors who had the mistaken dogmas still ruled. But before we plunge into that part of information theory part, let's just talk about the IT2, the technology avalanche, which basically tells us, alerts us that, look, there is something really huge coming. So whether we are ready or not, that's a big question. Essentially, this beautiful picture that Zoltan helped me with pictures the story pretty well. We know that these things are usually very solidly built. I wouldn't worry about technology, especially not in Google. if there was any kind of challenge in the history of computation technology, computation technology rose up to the challenge. And I'm pretty confident that this will not be such a horrendous problem as the other problem that I'm not sure about the wisdom of this fellow, because I wouldn't stand here. Because this thing, no, I'm not sure. The building will probably stand, but this is probably not the right mindset to risk it. And that pretty much tells the story that as far as technology is concerned, we are really strong and we can be confident, but we have some big problems that do we really understand how it will come around and so forth. Now, this is just reinforcing the same thing, especially when we have this challenge with these kind of logos, which of course are not my making, I just assembled them. One can really trust that if and when these kind of forces engage, we are in pretty good hands. And I'm happy to say that, not to disclose because it's very well known, that both of them have been engaged big time. Now, the key to genome informatics is, of course, computing, and I select the two markets. One I call the get info, which is the sequencing part, and the other is the use info, because why get the sequence if you can't use it for anything, any useful purpose? So both of these sectors of the market, they require heavy, very heavy computational challenge. The present day sequencing machines, they pass several times over the whole genome. This is a relatively long base, but it has to be assembled, and the assembly actually is the hard part, requires a lot of time. Now, in the use info sector, we already have personal genomics, pharmacogenomics, applied in bioenergy and biodefense, but we need advanced computing, for instance, for speed, because in biodefense, if you calculate some defense countermeasure within 2 months, but you need it in 2 minutes, then you have a problem. Now, also, it's already known, that very well known, that this is a very lucrative business. As early as 2006, Sirna, which was a tiny company in San Francisco, about 15 people, mining microRNAs, was picked up by Merck for the sum of $1.1 billion. So we know that it does work, and it does deliver, and it does deliver even for big pharma. Now, this summer, on July the 14th, something very important happened. There is a DNA sequencing company in Menlo Park, which is called Pacific Biosciences, and I do recommend looking up their webpage. You will have some spectacular technology on the bottom of the page. They got an investment of $100 million from Intel Corporation, associated with the really top-notch venture capital company league. So since that time, we know that Intel is in it. Actually, they are in it for a double dipping. They will manufacture the chip, which sequences. And then when there's this avalanche of data, because of the cheap sequencing, it will be less than $100. Then, of course, they will, again, profit on the processing part. Now, this is probably the key image that describes the present-day status. This is time, and this is the cost of getting the sequences, and this is this is the amount of information that arises from the sequences. Now, here we are today in 2008, just about wrapping it up. Applied Bio just announced that they can do a full sequencing for $10K. Complete Genomics in Sunnyvale, they announced that they will do, in 9 months, they will do a complete human sequencing for $5 grand. So far, there are 9 particular individuals sequenced in the world. And you can imagine that as the price drops from the present, suppose it's 10K, it used to be a little earlier 50K, and it was $1 million, it was 100 million. When it will drop under $100, Pacific Biosciences announced it for 18 months from now, they will sequence the whole genome in 4 minutes. Then this will be the time, probably around that time, maybe everything happens earlier, like in the internet. When you go to your annual, you will have a blood test, and you will also have a full DNA sequencing test. And if you are, your doctor suspects that there is some growth on your skin, then it will be possible to do a biopsy, and within 4 minutes, sequence that whole growth and establish the exact diagnosis, whether it's malign or benign, and what kind of therapy should be used, individually tailored to your genome. And then we will go further on this. This will kick in probably around this time, but somewhat later there will be a genome-based economy that we envision. Genome-based economy is that consumer choices will be guided by your personal genome. You will have your personal genome on a stick and maybe on a barcode device. I will refer to it later. And then you go shopping. And you buy that kind of stuff, whether it's food or whether it's medicine, which is really cut out for you, for your genome, not for somebody else. Because we all know that each of us respond to different foods and different medication in a different way for a very good reason. Oops. Now, as of October 6th this year, Kevin Davis, a very well-known editor of the BioIT, he noted that Complete Genomics in Sunnyvale will not be selling the individual sequencers, but they will offer a service, which is interesting. And especially it's interesting when he said, the company's CEO said, that we are putting in a Google-style data center to manage the data. Now, I don't know. That should be very interesting from Google's viewpoint. Also, it should be very interesting what kind of IT technology infrastructure they are going to build. And there are some signs that the infrastructure which is being built, actually, they are putting it together right now as we speak, because it's only about 9 months that they want to sequence, that it may or may not be the best choice. Now, there are some new toys. Actually, not new toys. These are devices. One of them is called the DRC plugin. It looks like this. This is an actual copy. And we are fortunate to have, as the CTO and founder, Steve Kasselman in the audience. And please talk to this very nice fellow. And he's widely known as the father of reconfigurable computing. The big thing about this is that it has a parallel chip, an FPGA chip. And the FPGA chip as a data flow machine can be reconfigured on the fly in a very short time. And therefore, it adds parallel processing capabilities for the sequencing, and as well as the downstream data analysis. Now, of course, that's not the only company which does this. This is Extreme Data. Extreme Data is close to Chicago, and they don't necessarily focus on genomics, but financial computing and, of course, defense computing used to be a traditional market for parallel computing. And even the parallel, the FPGA device sitting on their plug is different. In this case, this is the Altera. And in DRC case, DRC's situation is the Xilinx parallel chip. Now, you may note that actually it plugs in to the socket of the serial CPU. So if we have a machine with several of these traditional CPUs, you can just take one out and put a parallel processor in, and then you have a hybrid computer. So this is pretty much the future, as we see. And there are several other companies, but these two that I pulled are the lead ones. And of course, the big thing is speed, because the parallel FPGAs provide typically 100x acceleration, sometimes more, depending on the algorithm. And but also, by the way, their power consumption is extremely low. So it's green computing. But that's not the main thing. Now, in a very large, a very broad perspective, we have all of this ecosystem of all of these, the DRCs and the Extreme and the Intels and the HPs and the Xilinxes and the Alteras at a different level, like a parallel processor, serial processor, integrator, parallel processor, serial processor, integrator. There are about at least 16 combinations how to come up with a genome computer. And there is a horse race on among all of these players and more. This is just a few. There are more that I could mention, which actually will elevate us into the age of genome computing. And genome computing will be this kind of parallel hybrid devices with a genome operation system and killer applications designed for genomics. This is where it's at. So we are very similar situation like we were with the PCs back 20 years ago or so, 25 years ago, when there were all kinds of combinations. We don't even remember some of those, the Commodores and the other hardware associations. The genome OS and the killer applications, in that case, the PC OS and the killer applications were done by Microsoft. And actually, that era was won by that company. So here from this turmoil, what we will see as the genome computing emerging with a possible prize of genome OS and killer applications. Now, this is for technology, but now I have to tell a little story about the other side, which is the information theory of genomics. And 2007 was a very big year in genome informatics. There was an explosion of information. In Nature and in different other journals, Francis Collins, who actually led a group in NIH, they came up with a 4-year study, essentially saying that the scientific community will need to rethink some long-held views because they were wrong. And then he resigned. And they're playing guitar and riding a motorcycle. The NIH head, Zarhuni, also resigned. So basically what is happening, that a genome informatics explosion meant that the gene is a myth. This junk DNA is not true, it's junk. It can also be functional. The genomes of you and me are individually different. And the genome in me or in you, for all your or mine 100 trillion cells in a single human, are different. They differentiate. So there are this many gigabases in one genome, 7 billion people, and 100 trillion cells in each. Genome information exploded over 25 orders of magnitude. Now, of course, in Google, it's not a big deal because they talk about 100 zeros. So 25 zeros, they can probably handle, they think. But for genomics, this is something mind-blowing, because not only that they couldn't really look at the 40 megabytes of information in a meaningful way, this just blows them away totally. And worse, the information theory of the genome function is worse than non-existent because it exists, but 2 basic axioms are false. And gene regulation, as you can see here, this is March, Science magazine. It's very rarely addressed in informatics. So this is a exploded view of DNA regulation. I would say some assembly is required. We are not done with genome regulation from a theoretical viewpoint. So it's the information theory of genomics is pretty much at the stage where quantum mechanics didn't exist yet, but it was known that the atom would be splitting. So we have to create that kind of new science. This is, of course, the science core of hologenomics. Now, a couple of words about this totally wrong axioms. This is a hand-scribbled version by Crick in 1956, over half a century ago, when he scribbled and then published that DNA, which is transcribed to RNA, and then it builds proteins, it does work, but he says that it never happens that the protein information can ever go back to DNA. And of course, it's not a wise thing to say never. You should never say never. And especially when later he published this. This was a republication in 1970. Again, DNA and then RNA and protein. Protein is a dead end. Now, his friend Watson also joined in to try to save the boat, saying that the DNA and RNA and protein is there. I think he was way too clever not to forbid the recursion. But if you insist on this very straight line, then you have a problem. And of course, Ono said that why recurse if you will only find junk? So this shows that the fundamental axioms are just plain, totally wrong. So I can picture the situation. We are lucky because this is, of course, from the classic picture of Anderson that the emperor has no clothes. Now, I'm very happy that I don't have to say that the emperor has no clothes because all the emperors are dead or retired. This is why I could publish the paper, which said that there is recursion in the genome. And of course, recursion is something that is not new for anybody who knows anything about technology and engineering. So basically, this was the problem. The black figure is Crick's figure. And then central dogma prohibited the recursion. And then Ono said that even if you do go back, there is no information, there is junk. So therefore, there was a double barrier which was there. And by yanking them out, saying that, well, it's not true, what do we have? We have a DNA, RNA protein, and DNA, RNA protein, and DNA, RNA protein. Continuous recursion is an iterative recursion. And I published it this summer, as I said. Now, it means something theoretical, but it means much more. It also, I call it sometimes, for lay people, I call it the circle of hope. Now, why is that? I will explain it later, but here I'd like to visualize this transition from the old paradigm. This was on the 50th anniversary of double helix. Then a Scientific American cover story was this, that there is a double helix and there is a gene. And then, of course, according to the wisdom at that time, if a gene is active, then it develops some, for instance, a protein structure like a brain cell. This is a brain cell. In my case, it's a Purkinje neuron. It doesn't matter too much. So the gene paradigm was a forward growth paradigm. Now then, in hologenomics, this is redefined. This is OK. This is true that there are genes. Nobody says that genes are non-existent, although the definition is changed quite a bit. But then the big thing is that the growth is recursive. So then if there's a gene information. It generates a fractal template of like a little stem of this cell. And then this fractal template actually recurses, the protein recurses to DNA. That was forbidden. No, it's not. Why does it do that? Because it picks up information from what used to be called junk. And not only it picks up information from the recursion, but it also puts in a cookie. Now it marks that information, this secondary information, that it has been used, so that you don't want to use it again. And then this is called methylation chemically. It doesn't matter too much here. And also microRNAs are involved in this process, but I don't dwell too much on this. Now the rest, of course, is almost automatic, because in the second step, with that auxiliary information, you build this fractal template further. Now you can see that this template actually appears here and also here. This is the Lindenmayer fractal growth, and that growth is supported by this iterative recursion to the genome. And then, of course, you go a couple of steps, and then Gloria asked the question that, now, how does it stop? Oops. Okay. How does it stop? Now, growth, as we see here, growth has to be supported by information. And if you run out of information, then you recurse, but you don't find any more auxiliary information, then you stop the growing. This is why the trees don't grow to the sky normally. Now, of course, sometimes, is this the end? It's not the end. This is the beginning. But because connecting the dots after half a century, there is a recursive algorithm which opens to information science and technology. But also, you can see that if something is wrong with these cookies, with this methylation, for instance, this methylation didn't occur in the right way, then this already grown Purkinje cell can recurse, and oops, I can use this information because it has not been invalidated. And then it grows into even more complex something. It's called cancer. And in cancer, the methylation process, which inhibits the reuse of this secondary information, is known to be a very important factor. And of course, likewise, if there is something wrong with the methylation that a recursion would like to find that auxiliary information, but it's not there, there is some methylation problem there, it's already methylated, then the growth is stunted before a full development. So in the recursion, we know the mechanisms of several very important pathological processes. Yeah. Now, if we talk about diseases to medical doctors, then they would say, oh, gee, it might make sense. But for us, who know about information technology and information theory, and we know if we are physicists like Zoltan, and I also hold a degree in that, this recursion means that the second law of thermodynamics doesn't apply to the genome, because the genome system, by proteins interacting with the DNA, makes the system open. And that's a big thing, because if the DNA RNA protein is closed, then the increase of entropy is inevitable because of the second law of thermodynamics. And that is a horrendous problem. But with the circle of hope, proteins are what you are and what you eat, and what your grandmother didn't eat. They can interfere with the DNA open system. Now that is hopeful. Why? Because watch out what you eat. Because it may be very good for you, or it may be very bad for you. Actually, it can alter your genome. And that means that genomic entropy can be defeated. And also it means that you are not stuck with your genome glitches. So if some of these tests will show that you do have some genomic glitches, it doesn't mean at all that you will have an incurable disease. You might if you don't care. But you make preventive measures, then possibly you don't. There is hope here. And the circle of hope was strong enough for Mr. and Mrs. Broad to donate $600 million to create the Broad Institute of their own money. Why? Because a family member had Crohn's disease, a genetic condition that was considered incurable by the medical community. Mr. and Mrs. Broad, they got their wealth, about $7 billion from construction business. And they were just irritated that a bunch of doctors would brand that we could do nothing with this. And they also had some reasons to be encouraged by scientists like me and others. So therefore, he said, OK, let's do it. And you can know that the Broad Institute integrates Harvard Medical School, the old-fashioned doctors, and then but forces them to work with MIT. And we all know that in MIT, the T stands for technology. So it's happening. And this hope is not just hope. That hope is also venture philanthropy at this point. So the big pharma supports us. The big information technology companies support us. And even those people who really don't want to see that something is branded incurable, theoretically, can't do anything, when the science shows that actually the system is open for interaction. Now, this is about Sergey. I would not go through this in a very detailed manner. The point is that, this is the point, that he said that I also have the opportunity to perform and support research into this disease long before it may affect me, because he has a higher proclivity that he may get Parkinson's later on. Now, here is something interesting. In a recursion, I not only say that it's a recursion, but I also say what kind of recursion it is. It's a fractal iterative process. Now, I have my means software, which is called the Fractogen Miner, which searches picnon-like elements about these short nucleotide strands. And you can see that they repeat self-similar repetition, except that here, the letter O, actually, there's a tandem repeat. There is no repetition anywhere here. But also, within the tandem repeat, there is a binucleotide run. Now, these are glitches that I can mine, and these glitches are much more complex, almost infinitely more complex than just SNPs. This is why I have done this. And I could show the actual sequences, but there is intellectual property here, so I would rather not. But the software is ready. And if we can run the same software that, by which I established this, this is actually for a Parkinson's SNCA promoter. It's not even a gene, it's a promoter of a gene. And we know that there are about 3,000 glitches analyzed for Parkinson's. This is known, by the way, from the Michael J. Fox Foundation. And they got $2 million help from Sergey Brin to establish this database. So things are happening. Now, the good thing is that now if there is hope that you can interact with the DNA by as simple means as regulating your protein intake, what kind of food do you take, it's very well known at this point. This is by Waterland. And these are genetically identical mice, genetically identical. And they have the same genetic tendency to overeat. So they should be, both of them should be fat. But this guy is fat, like he should be, because he's in a standard diet. And this guy is nice. Why? Because he or she, I'm not sure, was on a methylated diet. So the methylation that I was talking about in this recursion, now modulating that methylation actually can make this difference. This came out just a couple of months ago. It's a spectacular epigenomic study. And it's as simple as this. Watch what you eat. And of course, those people who supersize, they might get this. Those people who don't supersize, they might get that. Now, this is another paper I just found that the Chinese knew long time ago, for a couple of thousand years that this came out in a scientific journal about the methylation of tea catechins. Green tea has been suggested to have beneficial effects against many diseases, including cancer, cardiovascular disease, and Parkinson's disease. So it may be that even on a regular green tea diet, it does something good for you. That has been known for the Chinese without knowing anything about genomics, or not much, and certainly nothing about parallel computing. But there is a lot of wisdom already out there. Now, so this is where we are. We can already do diagnosis for patients. Like with this saliva sample, by probing the SNPs, we can tell whether there is a likelihood here or not. We can make them health partners for disease prevention. Now, for instance, in Parkinson's, if somebody says, oh, there is a red flag here, then the person goes into a prevention mode, trying to do the thing which might prevent the disease being a full-blown disease. Then also in the next step, it will be personal genomic guidance to customers, because there are choices made in this world, and very often you want to make sure that whatever you are opting is actually good for you, good for your genome, because our genome is personal. And this is the big vision, that in a genome-driven economy, the consumer choices will be to fit or fix your genome, making sure that they're as good as they can be, or may even affect, for instance, in the methylation part, that kind of OID recursion should run as smoothly as possible. Now, this is from a USPD application. I will not dwell on this, but this is how you are going to shop. Now, read the label. Good luck. There are too many data. So it's much better to have some handheld device, and then just click on it and see through the nurture server and the nature server. And of course, you have to have your genome computer, and also you have to have your personal genome sequenced, which plugs into here. And then on your computer, you can enter your preferences. Then you sync it with your handheld, and you go shopping. So that will mean that you don't have to read the label. And in fact, the label doesn't even say anything personal to you, because it doesn't know who you are. But through a system like this is what we are going to see maybe in a couple of decades, maybe sooner. Any questions? Before we have some questions, I will have some additional 3 slides for the recursion thing. Now, can you imagine, everybody knows that the internet is fractal, that in a Google environment, what algorithm could you do in Google if a dogma would prohibit utilization information from internet users? It pretty much sounds like a total bloody nonsense to prohibit feedback from internet users. This is how the internet search engine works. So in this environment, of course, this is another statement that the web is fractal. This is by Flake and Pennock from a research lab, which is not Google, but shall remain unnamed. And also, they say that we have seen through this book how circular relationships, circular positive feedback loops are the key to creation of fractals and chaos. And so it is on the web. So I'm hoping that in this setting at Google, things are coming together, and they come and they make sense. And there's nothing weird about recursion. There's nothing weird about fractals. There's nothing weird about trying to put a new science into solid theoretical background so that you can generate algorithms. Like this wonderful world wouldn't exist if the page ranking algorithm would not be so marvelous. That's an algorithm. And of course, it takes a few software developers, 10,000, something like that at this point, to make it happen. But it's very well worth it, and it's very profitable. And if we apply the same kind of knowledge and understanding in genomics, it will be good for you, good for us. It will be good for the economy, and it will be good for our health. So maybe in the meantime, you are wondering what kind of questions you want to bombard me with.
Well, I wanted to, so you're saying saying that the protein that you eat can have something to do with the genetics, or, with your gene and your DNA?
Yeah, Steve's question, Steve Kassaman, the father of reprogrammable computing from DRC, he asked me whether the protein recurses to the DNA, and therefore it interacts with that. It is a well-known fact. It's not my making up. There are literally hundreds or maybe thousands of papers actually showing that it is happening. So it's nothing pulled out of thin air. In fact, nothing in this presentation is pulled out of thin air. This is very serious research in a stealth mode, because there were 2 axioms actually blocking my way. I never believed in either of these axioms, but I figured out. Now, suppose that they are not true, then what? So I developed the fractal algorithmic approach, and of course patented it in 2002. And then whenever it was possible, like saying so in a scientific journal without the scientific journal blocking your way, I could say it because the old guys passed away or retired, and they no longer care about it. And it's the facts are there.
So what's an example of something that you might eat that would protein with feedbacks on how?
Well, I would suggest to you to go to a Chinese. Scott asked whether there are some other examples for proteins that you might want to take. 2 suggestions. We know that the vitamins, they are called vitamins because they are vital amino acids. This is how the name came about. And amino acids are the components of proteins. And you know damn well that if you don't have vitamin C, then your teeth fall out. So you might as well eat green leaves, or lemons or whatnot, or just take a multivitamin pill. Many of these genomic diseases, including Parkinson's, in Parkinson's, the vitamin D regulation is way off. So some of these diseases can be not cured, but totally made symptomless by massive amount of vitamin D, but not the regular doubling it like when you buy the multivitamin supplement, but about 100 times of it, because the system just can't, it ruins it, basically. So vitamins are one thing that you want to check out. The other, and of course, they are very personal. The other thing that you want to check out is go to any kind of Chinese herb store. And you will see herbs that you have never seen before because they have hundreds or thousands of them. And that old guy, he knows that, take that tea or take that whatnot, and that's good for your this or this or this or this or this and this. They know it by experience. They don't have the science of it. They don't have the technology of it. And we are not yet there that I go with my barcode reader and I click on it, and they say, okay, this is there. But I should say, I should have to rely on the Chinese guys' wisdom. But every protein has the possibility of helping you, or everybody knows about mad cow disease, which is deadly. And mad cow disease is a classic and a proven example of a protein coming from the mad cow to your brain, killing you, no cure. So there are all kinds of positive and negative examples, by the hundreds.
You're saying, though, too, that because of recursion, my genome one day is doing this and needs more of this and less of that, perhaps. And then tomorrow, I might need a lot more vitamin D, for example, right?
Susan Matic from Gutenberg public relation firm, she asked the question, whether in different stages of our life, as this recursive growth, which we call life is growth, we might have different needs for different kind of proteins, sometimes more of this, sometimes more of that, or less of this or less of that. Of course. Now, we all know that because I think all of us, we are adolescent. So we went through the puberty. And we all know that in puberty, certain things happen to females and males. And there is a surge in that stage when within a year or two, there are spectacular growth and other changes, including your brain, your emotions. And that kind of process, there are some special needs at that point. For instance, now if somebody, a typical thing is to have the acne in adolescence. And that is, of course, genomically programmed. Why at that age? Why not later? Why not before and why not later? So absolutely exactly. This is why already, if you go to Costco, you can buy the multivitamins. It's silver gray for 50 and older. Because the vitamin needs are known to be different in different ages. For instance, vitamin B12, which is a very important vitamin, is much slower to absorb at an advanced age. So you have to take more of it. If you are young and you take that kind of mega doses, that you will be so hungry that you will overeat because you absorb them all. But now if you are not that young anymore, then you need a higher dose because not all of it will be actually taken up, and then not all of it will be used. So absolutely and definitely, the genome is not constant. It changes all the time. This is why you die, because you run out of information which sustains growth, unless you are hit by a car. That's a different story. But a normal life of a mouse, why is it that a mouse, which has about the same amount of information, little less, but the genes are 98% the same as the human, why do they live for 3 years only? And we live, some of us for 100, some of us 50 at least. Why is that? That's programmed into the growth process, because the growth process needs to pick up the information to keep the growth fueled. And how fast, what is your clock speed? Now, a mouse has a pretty high clock speed. It burns up all of this information much faster. So it gets into adolescence, I think, in a couple of months. And then probably 3 years or so, they're dead. Yes?
You started your presentation with saying DNA is at the doorstep of IT. So what are the limitations of IT? Why do we need to look at ultra-xylins and intel and AMD?
Okay, yeah.
Speedel?
Yeah, Gopi Rangan from Altera is asking this question from a very techie viewpoint, because he's a very techie guy. Why, when I say about information, why do we have to mess with hardware that I showed one example of, and of course Scott can show a much more recent model. Why? Well, we like to do that. For instance, I have a degree in engineering. So I also have the mentality that we have to understand something, then we have to build something so that you can use it yourself or sell it or make other people happy with it. So here the information, the understanding is not enough, because you can be as smart as, I don't know, but if we don't use that knowledge, then we can't help those people who are dying. Because there are hundreds, if not millions or 100 millions of people actually dying of diseases which are branded junk DNA diseases, just because doctors were prohibited by a dogma looking at that vast majority of the genome because it is called junk. So this is a life or death issue. And here we need the computation. For instance, if there is a growth and there is a biopsy, now do you want to wait a year to establish whether it's a melanoma or it's a very mild form of skin cancer? No, you don't. You can't afford. You want that kind of diagnosis as quickly as possible. So you have to go for speed. So in sequencing, some of this sequencing as of today it lasts 3 months at least. It used to last, the first genome was sequenced in 4 years. Now, typically, Jim Watson was sequenced in about a year. And as of today, the state of art is that it takes about 3 months, mostly of computer work, assembly of these shotgun pieces to put them together like a jigsaw puzzle. And it just takes too long, and also it's too expensive. If it's faster, then it's less expensive. And of course, I can deliver you the personal genome. This is why Intel said that, okay, let's go for it. But Intel will only produce the sequencing chip and the serial chip. And the process is, as you could see here, the genome itself is a massively parallel processor. Not just one gene which acts, all of them act. It's more like an orchestra with all the instruments, all of them acting, but some more and some less, and very dynamically. So that kind of massively parallel processor, the genome, requires its match in information technology so that we can deal with this beast. And this is not an intellectual curiosity whether there are aliens who are similar to us somewhere in the universe. It's interesting. Whether there is water on Mars, that's also interesting. But I don't care too much about that. I care a lot more if I have at least 2 genetic syndromes. One, male pattern baldness. It's known to be a genomic syndrome. And also I have a pointed ear. That means that 1 million years ago, I was probably a fox. And it's still there. But I may have some others. And thank God I don't know that. But and one day it may turn out that melanoma showed up, which can kill you in 6 months or less. So this is an issue that we ultimately care the most about, our well-being, and also the well-being of our loved ones. Also, medical technology. And if you don't make it fast enough and cheap enough, then we can't maintain the healthcare system. Further questions or follow-up? Now, of course, for people like Gopi and others in the FPGA side, now they cannot do it alone. This is stuff that requires companies like Google. Thank you very much.